Hello everyone, this is Dr. S and welcome to Biochemistry. Here my goal is to introduce you to the course's learning goals at large, and we'll talk a little bit about course formats. And once we cover that, we'll be jumping into our first topic, which is entropy. And I like to begin this course with a quote from a novel written by Douglas Adams called The Restaurant at the End of the Universe. This is the sequel to The Hitchhiker's Guide to the Galaxy. And the quote is over here: "There is a theory which states that if ever anyone discovers exactly what the universe is for and why it is here, it will instantly disappear and be replaced by something even more bizarre and inexplicable. There is another theory which states that this has already happened." So, at our own peril, let us continue. Although before we dive in, I think introductions are in order. I myself grew up in Long Island, New York, Suffolk County, and for my undergrad, I attended what was known as St. Joseph's College at the time. And while I was studying a degree in biochemistry there, I first got into research because I was also working part-time at Brookhaven National Labs, which was nearby. And it was an absolutely fantastic work experience. I caught the science bug, and I decided I wanted to do this full time. I went to graduate school at Emory University in Atlanta, and it was so hot there. And besides sweating very often, I spent most of my time there studying protein nanotechnology, essentially. Specifically, these complicated networks of a protein called amyloid beta. And over here, I have a video, a simulation of its complicated molecular structure that I spent many years trying to resolve in that simulation. And after I defended my PhD, I then moved on to Cornell University next, where I studied something completely different. There, I was doing total synthesis, essentially. I designed and made this hula hoop looking molecule that was designed to trap and recycle certain precious metals like europium and cerium, which are, for example, in the phosphors on your monitor display that you're currently looking at. Once I concluded my postgraduate studies, I got an industrial job at Stony Brook's School of Medicine, and I worked there as a radiopharmaceutical chemist, which was a lot of fun. My job there was basically to make these short-lived radioactive drugs in the morning that clinical researchers would use to image the brains of patients that had enrolled in various studies. You know, sometimes we looked at people with Alzheimer's disease, sometimes we used it to study the brain of sleep deprived people. And as much as I loved doing science in a clinical and industrial setting, I really missed teaching. I actually taught all throughout my graduate studies. And so what I did was basically teach night classes while I would work during the day as a practicing chemist, you know, just to keep that part alive inside of me, I suppose. And I did that, taught at night, worked during the day until it became possible for me to do the switch and become a professor full time, where I started uh, my position at Malloy University in 2023. So that's me in a nutshell, I suppose. The first point of order I'd like to bring to the table is our course objectives. This is copied and pasted straight from the syllabus, so I won't waste your time reading it to you here. I am, of course, happy to go over any questions you have about the syllabus or really any of the contents. Please never hesitate to reach out. My job is to make this as clear and painless as possible. And if you've taken a science class before, you know that sometimes it can be anything but those two things. Next, I've shown here the specific learning goals, or the learning goal rather, for this lesson's content. And the theme today is entropy. So by the end of the video, you should be able to explain how biological systems can spontaneously order themselves without violating the laws of thermodynamics. And you'll be able to make sense of that in just a few more minutes. I want to say, though, that biochemistry can get a little overwhelming at times. So when you're studying and you're not sure where to start, my recommendation, go to the first slides of each lecture. I will always post the learning goals, and I want you to review them and ask yourself, honestly, have I met these learning goals? If not, well, this may be a good place to start your reviewing. There's absolutely no shame in pausing or rewinding or rewatching bits of these videos because there's a lot of material to cover and it's quite advanced. I know I did when I first learned the subject, and I think you'll be most successful if you take your time with it and be patient. Another thing to know about my lectures is right after the learning goal slide, I will have a class opener type question, which I call the molecule of the moment. The purpose of this is to get you all in an empirical mindset so you're ready to address today's learning goals and get your brains all warmed up for the challenges coming ahead. And these are going to be usually rather difficult questions. Sometimes they find a way of reincarnating themselves on the exam, sometimes word for word, sometimes with a few tweaks, but I want you to take it seriously. And to help you do that, I'm going to, twice a week, have a random student attempt to answer the question before the class. And if you get that question correct, you will earn extra credit on the next exam. If you are not lucky when it's your turn, don't worry, I'll have enough of these to ensure that every student gets a shot at winning the prize points. Keep in mind, though, that that does not mean I will explicitly answer the class opener question in the video. That is something that I need to see you show the work on. If you pay attention, though, sometimes the answer can come out organically or implicitly somewhere down the road in the video itself. So the power's in your hands. With all of that aside, I will be presenting to you the first class opener question, and I'm going to keep this one on the simpler side. Your goal is to list the 7 characteristics that are found in all forms of life. Because this is the first one, I decided to be kind, and I gave you gifts that represent the answers, since general biology may have been several years ago for most of you. I highly recommend, though, that whether or not you are selected to do the question, pause and try to answer it on your own. It's a good method of studying regardless. From the list of 7 characteristics seen in all forms of life, the one that is possibly hardest to wrap your mind around, but most integral to biochemistry, in my opinion, is order. Biochemistry is all about order. This is a Dr. S opinion, but I will die on that hill. Think of it this way. If I took an organism and I disordered it by, I don't know, putting it in a blender, do you think it's going to survive the disordering treatment? I don't think so. So it's very hard to talk about order without mentioning its opposite, disorder, which can be thought of as entropy. You might recall way back from general chemistry that systems proceed from order to disorder spontaneously, or low entropy to high entropy states. This is why your room gets messy all on its own, and you have to put work in to clean it or reorder it. All right, well, why is that the case? The equation over here, S equals K times the ln of W, describes that the ways of a system that can be rearranged is proportional to its entropy, or its potential to dissipate energy spontaneously. This is known as the Boltzmann equation. And the term S represents entropy. And the units, if you're curious, is joules over moles times Kelvin. K, the Boltzmann constant, is a proportionality constant with a value of 1.38 times 10 to the negative 38 joules per Kelvin. Very small number. And W is the confusing one. W is a term that represents the number of ways a system can rearrange. For the purposes of this course, this is not an equation I'd expect you to whip a calculator out and solve. What I instead want you to do is look at the equation as a story. It's trying to teach you something. Let's apply this to a simple system like a glass ornament, specifically a glass ornament that got knocked over and shattered on the floor. A glass ornament is a covalent organic framework of silicon-oxygen bonds, and when it shatters, all of those bonds, or a large proportion of those bonds, I should say, breaks randomly. It flings the glass shards apart. We get potential energy converted to kinetic energy. W is huge for this system, because if I dropped a million or a gajillion identical glass ornaments, they're going to fracture in a million or gajillion unique ways, most likely. Since we can break it in a gajillion ways, W is going to be huge for the process of breaking the ornament, which means entropy is going to be large for breaking that ornament. Now let's consider this equation down over here. Diatomic A reacting with diatomic B to form two equivalents of the molecule AB. This reaction is an equilibrium. The only other accessible state besides the product state is the reactant state. So W in this instance is not a big number. It is in fact 1. If we plug this into our Boltzmann equation, the ln of 1 is 0, and any number times 0 is 0, therefore entropy is 0. The key takeaways is that S equals 0 for reversible processes, or processes in equilibrium, and S is big, greater than 0, for irreversible processes, like dropping a glass ornament on the floor. All right, that's nice. What does this have to do with life again? Well, for starters, it implies that life is far from equilibrium. Living creatures, even biomolecules, are more complicated, in my opinion, than a simple glass ornament. And breaking that glass ornament is going to release a lot of energy. Where did it get that energy in the first place? You break a glass ornament and you'll never be able to have it unshatter magically. If you want to remake it or fix it or get a new one, that's going to cost the universe energy in some way. The glass worker, for example, must spend energy to melt it back together and make a new one. It has to burn a furnace, use your muscles to lift the materials and put it in the fire. And all right, well, where did the glass worker come from? The glass worker's mother had to make the glass worker first. And the glass worker's mother had to be made. This is getting quite philosophical. So, this brings us to a key question to life and biochemistry. If biochemistry is all about order, making beautiful, intricate systems like glass ornaments, and even more complicated than that, how does it happen? Order isn't spontaneous. Our rooms will never clean themselves. So the question that kind of keeps us up at night is, how did life acquire order in the first place? What is the origin of order? I'd be lying to you if I said that biochemists knew where all the order came from that made biology possible. However, we do know where some of that order comes from. And part of biology's order stems from the macromolecules that compose us. I'm talking things like proteins, nucleic acids, lipids, and carbs, everyone's favorites. These are all highly intricate molecular compounds, and their highly intricate structures dictate their highly intricate functions. The literal shape and polarities of these molecules is what keeps us in one organized, intelligent piece. The molecules of life are actually the instructions of life. And I don't just mean DNA. Yes, DNA is the code of life. No one is ever going to tell you otherwise. And the order of nucleotides is certainly important to having a cell function. But think about lipids. We don't normally think of these as ordering entities, perhaps, but lipids are crucial for compartmentalizing the cell. They define the boundary of what's inside and outside of the cell, or what's inside and outside of the organelle. If you get rid of those compartments by putting the cell in a blender, guess what? No more life. You see how important it is to have low entropy, aka high order? Another great example is structure. That is a kind of order, a kind of instruction that biology gives to certain proteins like collagen. The job of collagen is to hold your connective tissue and skin together. And it can do that all thanks to basically 3 amino acids on repeat. You change one amino acid, one letter in those instructions, and then the instructions stop making sense. Collagen can't do its job. Your skin becomes more disordered, aka it falls off your body. That's called scurvy. If the instructions behind collagen get messed up in a different way, say through a genetic mutation, it can become too floppy and flexible and result in vasculature problems or dislocations. The word for that is Ehlers-Danlos syndrome. perturbing order can be the beginnings of disease and pathology. Here's a video that visualizes, for example, how proteins can contain instructions, like instructions for assembly, baked into their molecules themselves. This video on the right-hand side is a simulation of hepatitis viral capsids, which are shown as these blue pentagons swimming about chaotically. And if you squint your eyes and look, you may notice there's a single red noodle floating in the midst. That represents an RNA molecule. And if you watch the simulation play out, you can see that the RNA genetic material of the virus will combine with the capsids and automatically form a virion, a viral particle. How is it doing this? Well, there's essentially only two key interactions that drive this whole phenomenon. The pentagon-shaped capsid proteins have sticky edges, for example. They are attracted to each other, like so. And when they click together, they're going to do so at a slight angle. So as more of these capsids click into place, it is going to curve into itself until it forms a hollow ball or sphere. Meanwhile, the interface of those capsid proteins are highly positively charged. It therefore likes to bind highly negatively charged molecules, such as RNA. So what happens? That huge RNA strand will collide and grab onto several free capsids, and it's going to reel them all together and facilitate the formation of the virion. What we are seeing here is the RNA molecule working with the viral capsids to reduce the entropic barrier to assembly. This is what I mean by molecules having instructions. The instructions here, baked into the molecules, are very simple. Connect the pentagon edges together, and then connect the pentagon faces to the genetic material, the RNA. And these two simple rules is all that it takes to form a complex, highly ordered entity spontaneously. No ATP was used in this process. And I think this is amusing. As an aside, nature is fully aware, or not aware, that instructions get misread. This simulated capsid situation on occasion has accidentally formed conjoined incomplete virions, like we see over here. That's a mistake. And all right, maybe there were bugs in the software, it wasn't tuned very nicely. Well, it also happens in real life. Caught in the act, we took a electron micrograph of assembled virions, and voila, we can see two of them assembled improperly and conjoined as a result. This isn't the end of the world if it only happens a few times out of a billion, but it goes to show that the theme of biology is often don't let perfection get in the way of good enough. More importantly, though, I need to hit pause and circle back to the broken glass metaphor. I told you earlier that the laws of thermodynamics prevent us from getting something from nothing. We can't fix or build the glass ornament without some sort of energetic cost. Yet, it appears we did that here. The virus constructed itself spontaneously. But did it do that for free? There may not have been a glass worker or any other worker manually clicking these things into place. But was there a cost that we're not immediately observing in this simulation? Perhaps that viral capsid assembly example was an exception or a fluke then. I think when people hear about complex biomolecules coming together seemingly on their own, they think about the Miller-Urey experiment. This was a very famous experiment that was done decades ago. It was designed by Stanley Miller, and he wanted to know how biomolecules, how life basically could arise from not life spontaneously. And this is how he tried to probe that question. He constructed a contained glass apparatus and filled it with what we predict to be the components of Earth's primordial atmosphere. Things like methane, ammonia, carbon dioxide, very simple gases. And there's also water, which was heated up into the form of steam. And that steam, containing all those delicious, simple molecules, was passed through a chamber that was subjected to sparks. And the sparks replicated lightning, reducing power. Okay, and what happened next? Well, after we zapped the components in the electric chamber, we condensed it down back into a liquid and collected it in a flask. And a few days later, we analyzed what was in the flask. And what did we find? We found every single amino acid used in life. And I believe we even found the nucleotides as well when we repeated the experiment with more sensitive equipment. Getting complex biomolecules out of very simple, mostly inorganic building blocks is mind-blowing. And that reaction, by the way, is very uphill thermodynamically and also entropically. But it happens. And sure, we are supplying loads of energy into this reaction in the form of heat and electricity. But does that make it any less impressive? These aren't skilled glass workers here. It's random sparks, random heats. We've seen the cartoons where the bad guy gets zapped by a thunderbolt and gets reduced to ash, right? That is order to disorder. In the Miller-Urey experiment, we see the opposite. The ornament forms, the amino acids form. That's disorder to order. What gives? If that's not spooky enough, consider this. More recently is the work of self-replicating RNA molecules, which was discovered by a scientist by the name of Holliger. Holliger crafted a 96-mer RNA sequence that he called TC19Z. It rolls right off the tongue. And that molecule is able to replicate itself. You take one of these guys and you add it to a solution of free nucleotides, and it will eventually convert them all into polymers as well. And it will do this with a 1% error rate. No cell or polymerase or ligase is needed. It will catalyze its own duplication. And again, it's almost like the glass ornament is appearing to form itself. The RNA appears to order itself. Again, entropically uphill, but it happens. So, what the hell? Am I saying that biology found a way to violate the laws of thermodynamics? Of course not. It took us scientists a while to explain this, But Schrödinger of the Schrödinger equation had a poignant insight in his book titled What is Life? And in it, he said the following: life, contrary to the general tendency dictated by the second law of thermodynamics, which states that the entropy of an isolated system tends to increase, decreases or keeps constant its entropy by feeding on negative entropy. That concept of negative entropy, which we sometimes call negentropy for short, is what helps rationalize this paradox of order from disorder. We are not making order for free, because that would violate the second law of thermodynamics. Biology generates order, but only at the expense of creating more disorder, entropy, elsewhere. Organisms are order vampires. At least that's how I like to think of them. We steal a little pre-existing order, aka negentropy, from our surroundings to keep our own entropies low, and thus our order high or stable. And if we steal or take order from the universe, that is going to leave it more disordered than it started. The amount of disorder we create actually exceeds the order we steal. I know that's an abstract concept to wrap your mind around, but that will become clearer in the lectures to come. As for where specifically in the universe we steal order and how, well, you'll just have to stay tuned for next time.